Uno, dos, tres. Sorry, waiting to hear that ding-a-ling sound. Did I miss it? Ah, there's the ding. Okay, we talked about reefs. We've talked about life in tidelands, rocky coastlines and sandy beaches. Coming up on Monday, I want to flap my gums at you a bit about estuaries. We've already talked a little bit about seagrass beds and things like that. But okay, there's something going ding dong. Okay, there we go. But for today, I want to step back a little bit and look at things on a more global scale and look at what we call provinciality and biogeography, which is simply the study of geographic locations of organisms, the study of why you find certain species, where you find them. And there are some parts of the world, the land, the oceans, any environment you care to name, that have more species than others. You might have, I don't know, thousands of plant species in a swath of the Amazon rainforest, and maybe 10 or fewer in an equivalent size swath of Alaskan tundra or something like that. And in any given place, the number of species is a balance between species formation and extinction. New species are constantly in the process of evolving. Species might also be migrating into an area. And then in any given area, species may be going extinct, either globally, or we can talk about local extinction as well, where a species doesn't die out completely, but it's wiped out of a particular region. And the balance between the two is the number of species you've got in any particular area. How that plays out in the oceans is there's a couple of really big patterns. For most marine taxa, right, for most groups of related species, snails, clams, green algae, whatever it is we're talking about, the number of species present is greatest towards the equator and lowest towards the poles. So a bit of coastline close to the equator will probably contain many more species of snail, let's say, than the same amount of coastline closer to the poles. So the number of species is highest at the equator, and it tapers off in both directions as you go either north or south. That's called the latitudinal biodiversity gradient. The other thing is, when you look at continents, continents are to an extent separated from each other. I mean, you've got bridges between, let's say, Africa and Asia or North and South America, and you can have migrations of species between continents, but still, typically, an area as large as a continent will have sets of species that are related to each other and not that closely related to anything else. So there's distinctive animals and plants that are native to North America, native to South America, native to Africa, native to Australia especially. We call those biogeographic provinces. The oceans are a little bit different because the oceans are all connected to each other. You don't have the ocean equivalent of Australia, which is a continent-sized landmass that is not connected to any other continent, or South America, which is only connected to any other land by a very thin little strip. But you do get different regions of the ocean, and the boundaries here are not land boundaries. They're boundaries between water masses, and those create areas of ocean that tend to have species in common, and the species you find there are distinct from species anywhere else. And we call those biogeographic provinces as well. So even though the oceans are all connected into one world ocean, there are still some divisions within it and boundaries within it that give us the same kind of thing that we see on looking at land life on continents. You have big blocks of ocean with a lot of distinctive species that are different from what you would find in the next big block of ocean. And those blocks are biogeographic provinces. So here's some example. This is number of species of snail. The number of species of snail has been counted. On the x-axis, you've got latitude in degrees from a little south of the equator all the way to the North Pole. And the black line is number of species in the Western Atlantic, up the Atlantic coast of North America. And the open squares represent number of species in the East Pacific, the East Pacific coast of North America, from Mexico, California, Oregon, Washington, Canada, all the way up to Alaska. And you can see that the majority of species live in pretty tropical latitudes. The number of species starts falling off fairly sharply once you get north of about 25 degrees latitude. It's not at maximum diversity directly on the equator, and it might be interesting to know why, but you still get an overall gradient in that you've got lots of species in the tropics and progressively fewer once you get north of about 25 latitude. You get fewer and fewer species until you get all the way up into polar waters. This is data on marine copepods. You might remember that those are little roughly millimeter-sized crustaceans, little shrimp relatives. And each one of the colored dots on the map is the number of species of marine copepods that were found at 433 sampling stations, some of them on the coast, some of them visited by ships. So those colorful dots in the middle of the ocean are places where some ship stopped and took a water sample and collected a whole bunch of copepods and sailed on. And what I think you should be able to see is that you have the red and the orange dots are high number of species. That y-axis scale over there, that's not raw number of species. That's some kind of statistical measurement of it. But the more diverse sampling stations with more species of copepods are red and orange. In between is yellow and green, and the least diverse areas, the ones with only a few copepod species, are turquoise and blue. And if you look closely, you should be able to see that there's more red and orange in tropical areas, and there's more teal and blue up in polar regions. So overall, the pattern seems to hold in most places that we've got a latitudinal diversity gradient. More species in the tropics, fewer species towards the poles. If we take the same data and we regraph it, that graph up there at the top is the same data that you saw on the map. It's just plotted in a different way. And there is a fair amount of scatter, but by and large, the pattern holds in that average diversity, number of copepod species is greatest at the equator and tends to slope down as you get closer to the poles. In the middle, that's ocean temperature. And of course, that peaks at the equator and slopes down as you get towards the poles. Salinity, right there at sea, is actually highest a little bit away from the equator, because right at the equator, remember, you get lots of rain because of that intertropical convergence zone. Somebody's mic is on. Okay, somebody's mic's on, folks. Somebody's mic is on and they're playing music. Okay, it's a pretty good jam. Okay, somebody please mute your mic. Anyway, you might remember that right at the equator, you have that intertropical convergence zone where there's high rainfall. And hang on. Okay, Blake Mitchell, please mute your mic. Let me see, do I have the power to mute you? Okay, I don't have the power to turn off your microphone. Okay, yeah, I don't know what's up with Mike today. Okay, I'm just going to try to keep going. I don't know if I have a way to. I don't know if I have a way to signal him. Okay. All right. I've muted him on my end. So, okay. Hopefully that should fix that. Right. Okay. So yeah, the salinity at the equator is actually a little bit lower than it is at about 30 north and south on either side, which is what we'd expect. You know, we were talking about, you know, salinity in the oceans, and about rainfall being highest at the equator and lower at 30 north and south because of those Hadley cells. That's the pattern we should see. So copepod diversity is pretty strongly correlated with temperature. It might seem intuitively obvious that the tropics are more diverse. I mean, you've all heard of tropical rainforests and tropical coral reefs and things like that as being very diverse habitats, you know, as opposed to, you know, there's not a lot of things that are capable of living up on the tundra. Actually figuring out why this is the case is still a bit of a puzzle. I mean, it may seem obvious, but trying to come up with really good reasons for why it should be so is interesting. The rate of speciation may be higher in tropical taxa, although I'm not quite sure why that should be the case. I'll talk about the Red Queen hypothesis in just a minute, but it's possible that biotic interactions are stronger in the tropics. The ways that species can interact with each other. One might be a parasite on another, one might be a symbiont on another, one might eat another, one might compete for resources with another. It's been proposed that if those interactions are stronger, and if each organism is interacting with more species for better or for worse, that might drive speciation. And then the third possibility is the tropics may just be more stable. For about the past 1.5 million years or so, Earth climate has been cycling, and we've gone through 4 major stages of global cooling called ice ages. The last one came to an end roughly 12,000 years ago. At the height of the last ice age, we had glaciers covering North America as far south as Des Moines, Iowa. They didn't get quite down to Arkansas, but yeah, we had polar ice covering most of Canada and coming well into the United States. Similar things were happening in Europe up until about 12,000-ish years ago. That's going to have a big effect on biodiversity because a lot of species can't live where you've got solid ice. That's certainly a problem on land, and it's a problem in the oceans as well because sea ice can grind against coastlines and make it impossible for your typical rocky shore species to live there. And so the tropics may just be a more stable place, not subject to extinction caused by climate cycling over the past 1.5 million years, maybe. So about that Red Queen hypothesis, this guy's named Lee Van Valen, not Van Halen. That's a different guy. Sorry, pouring out a beer on the curb for Eddie. Lee Van Valen devised what he called the Red Queen hypothesis. It's called that, by the way, because all right, you've heard of Alice in Wonderland. Lewis Carroll, who wrote Alice's Adventures in Wonderland, wrote a follow-up book called Through the Looking Glass. And in Alice in Wonderland, part of the book is set in the middle of this weird game of cards, right? There's a king, there's a queen. They're based on, the design of playing cards. There's that weird sequence where the playing cards are bending into croquet arches and people are playing croquet with hedgehogs and flamingos. Through the Looking Glass is set in the middle of a weird chess game. The pieces are white and red instead of white and black. And there's a bit where Alice encounters the Red Queen, who starts running, and Alice has to run very fast to keep up with her. That's what they're doing here. And when they stop running, they're exactly in the same place they were before. And Alice says, where I'm from, when I run, I usually get somewhere. And the Queen says, well, that's stupid. Here, you see, it takes all the running you can do to keep in the same place. And that's why Lee Van Valen called this the Red Queen hypothesis. He argued that the main thing that makes species evolve is biotic interactions among species. Every species is subject to predators. I mean, maybe not killer whales. They're not preyed on by anything except SeaWorld. But almost every species has predators that are trying to eat it. Every species has pathogens, bacteria and viruses that'll kill it. Every species has competitors that will try to acquire the resources that it has. Every species has parasites. Every species has mutualists, symbionts that actually help it out. Every species is bound to lots of other species in its environment by these biotic interactions. The result is that every time one species adapts, that sets up change in all of the others. If a predator evolves tougher teeth, then its prey has to evolve a countermeasure, like swimming faster or thicker shells, maybe. If a parasite evolves some new way of taking advantage of a host, then the host has to counter-evolve some sort of countermeasure, some sort of change in its immune system, maybe, something like that. So every adaptation in one species forces everybody else in the ecosystem to adapt. And so each species has to constantly evolve just to keep the niche that it's got, just to keep ahead of the competitors, the parasites, the predators, and the pathogens. And we talked about this a while back. One way that species do this is by sexual reproduction. By constantly messing with your DNA, you generate variation, you generate change, and you generate the ability to evolve new adaptations to keep ahead of your competitors and predators and all that. So the idea is that maybe you have more biotic interactions and more intense biotic interactions in the tropics than you do in the poles, where there's just not as much happening. And that might explain the latitudinal diversity gradient. The problem is that some species haven't gotten the message. This picture shows a brachiopod. Brachiopods superficially look like clams. They've got a pair of valves that fit together that are connected with a little hinge and can open and can close. The fine details of anatomy are very different. And if you ever take, or you have taken, Biology 4401, I talk about the differences in a little bit more detail. Just know that these are the things that look superficially like clams, but technically are quite different in details of their structure. They're not that closely related to clams. They used to be much more abundant up until about 245 million years ago. They're very common as fossils in many rock layers up in the Ozarks, for example. But there's not very many of them now. There's maybe 300 species, but they tend to be more diverse at high latitudes. There are more brachiopods in, subpolar waters than there are in the tropics. And the same is true, for some reason, for red algae. So we have exceptions to this latitudinal diversity gradient, and I don't know that we have a really good explanation for why. So there, a lot of talk just to tell you that we don't really know. There's actually a number of taxa that show bipolar distribution, which means that you don't find them at the equator. You do find them close to the North Pole and close to the South Pole, but not in between, and they don't seem to migrate between. What you're looking at here is an organism called a bryozoan. A bryozoan individual is a little blob of jelly, roughly a millimeter across, with little tentacles that it uses for filter feeding. What's significant about them is that virtually all of them are colonial. You never find just one single bryozoan unit, but each bryozoan unit can bud off new bryozoan units. So what you're looking at, if you look at B, I'm seeing about 6 kind of oval or football-shaped structures. Those ovals or footballs are part of the casing. It's called a zooecium, if you really have to know. They're part of the casing that bryozoans build around themselves. And it may be a little hard to see this, but they're all connected to each other, or they were connected when the bryozoan was alive by little strands of tissue. One of them was the first to start growing, and then it budded off the others. So that's what you're looking at here. Each of those ovals is part of the little house that an individual bryozoan lived in. The species is called Calipora veslavski, and it's just an example of a species that lives in both polar seas, but it doesn't live in between. It's possible that it might have been more widely distributed and been able to migrate at some time when global climate was cooler. But right now it's in the Arctic and Antarctic and doesn't seem to be located in the in-between. So don't worry too much about exactly what a bryozoan is, but you do need to know bipolar distribution. These happen to be floating foraminifera. We've talked about these back when I was talking about plankton, that these are single-celled protists with an amoeba-like body. Many of these are benthic, but there are some that are planktonic, and these are the shells of two different planktonic species. And this is another example of bipolar distributed species that are found at both poles, but not in between. When someone looked at the DNA of these, they turn out to be very similar, which means that they split very recently. So maybe when the ocean was cooler, these were able to live at the equator, and then when the ocean warmed up, they moved north and south and are now separate. You've already seen this slide. You saw this in the context of evolution when we were talking about how new species form. And new species form when you get a barrier that takes a population and splits it. So you now have two populations that can't easily exchange members. So about 4 million years ago is when the Isthmus of Panama formed, connecting North America to South America. Incidentally, that allowed land critters to migrate in both directions. So South America ended up with jaguars and cougars and oak trees and bears. Those are all North American species that migrated south. And North America ended up with armadillos and possums and, oh gods, what else? Okay, those are the ones I can think of at the moment. Possums and armadillos, those are South American species that migrated north. That's what happened on land. In the oceans, you end up with what used to be one species getting split into two, one of which ends up in the Caribbean, like Strombus pugilis to the right, and one of which ends up on the Pacific side, like Strombus gracilior to the left. And we use that as an example of allopatric speciation. You can go back and read up on that if you're not clear. And it turns out that barriers aren't necessarily made of solid land. This is some groundbreaking work that was done back in the 60s by Jim Valentine, who was one of my professors at Berkeley and one of the most frighteningly smart people I've ever known. And these days, there's a lot of science that you can do by assembling data sets of lots and lots of numbers and then crunching them on computer. But Jim Valentine did this in 1966. When computing was still being done by punch cards. There's the coast of the North America. You can see Baja California to the south, United States, British Columbia, and then Alaska. He divided the entire length of this coast into blocks 1 degree wide. Then he used a bunch of published data to work out what species of mollusk, clam and snail, were present in each one of these 1 degree blocks. Once you've got that, you can use, if you have two areas that have species of something, you can calculate how similar the set of species in those two areas is. There's various ways of doing it. There's various, we call these similarity coefficients. The simplest one would be just to calculate the percentage of species that are in common. So if you have two areas with 10 species each, and 7 of the 10 species are identical in both areas, you can say these two are 70% similar. There's actually some reasons why you wouldn't want to be quite that simple. There's some more complicated ways that you can calculate this that, well, never mind. We don't really need to go into that. Let's just say that you can take data like this, where you've got lists of the species present in areas, and use that to calculate numbers that give you an idea of how similar each area is to the others. And again, we call those just biogeographic similarity coefficients. There's the Jacquard coefficient and the Dice coefficient. They're just numbers between 0 and 1 that tell you how much alike any two areas are. And so he calculated these similarities between the sets of mollusk species in each block. And there's his data. Okay, you certainly don't have to read all of this. Don't worry about that. Let me just show you the larger patterns. To the upper right, the upper right part of the square has a whole bunch of numbers written in those little squares. Those numbers are similarity coefficients, these numbers between 0 and 1 that tell you how similar any two areas are. Below and to the left, it's the same data, but instead of numbers, what he's got is shading. So, okay, if you have one block, okay, I can see the block numbered 40 down on the x-axis below this thing. If you find the number 40 and you go up, and you can compare block 40 on the x-axis to block 39, let's say, on the y-axis, and where 40 and 39 meet, you have a square that's shaded in very dark. What that's telling you is that the block of latitude at 40 degrees from the equator and the block of latitude starting at 39 degrees from the equator are very similar. They have almost all of the same species in common, or almost all of the species in one are shared by the other. Okay, I hope that sort of makes it clear. I'm kind of waving at the screen and forgetting that you can't really see what I'm doing. But yeah, the numbers in the upper right half are these measurements of numerical similarity. The larger the number is, the greater the fraction of species shared between blocks. And then the same data is found in the lower left half of the square. And what I want you to see is that, you would expect individual blocks to be, to have the greatest similarity with blocks that are closest to them, right? If you have one block of land, let's say it's the block of coastline at 40 degrees, that's going to have a certain number of species present. And you'd expect to find most of those species in the blocks on either side, right? Most of the species that live at 40 degrees also live at 41 or at 39 degrees, right? So you would expect that you're going to get more similarity in blocks that are close together. And by and large, that's true, but it's not even. You have some shaded areas that tend to clump together. So right in the middle, you can see there's a zone where there is some fairly deep shading. Latitude between about 35 north and 55 north, there's a lot of mollusk species that are found between 35 and 55 north, but they're not found north of 55 north or south of 35 north. And then north of about 55 north, you have another one of these highly shaded areas. So that's telling you that north of 55, the blocks tend to have lots of species in common with each other. but not so many shared south of 55 degrees north. So diversity is clumpy. And the reason has to do with the North Pacific Gyre. Okay, we've already talked about gyres, and you know you have this equatorial current flowing east to west across the equator. It turns north when you hit the Philippines, goes past Japan as the Kuroshio current, and then comes around as the North Pacific current. And it happens, this is not shown as clearly as I'd like, but it actually hits the North American coast at roughly 55 degrees north. And when it hits the North American coast, it either turns north and becomes the Alaska current, or turns south and goes down the Pacific coast of Washington, Oregon, and California. Okay, this is the California coast. Land is all shown in black. You know, land is not interesting. This is marine biology, damn it. And all of the colorful areas are water shaded with temperature. So cold water is shown in blue. Warm water is shown in, you know, yellows, oranges, and reds. And right at about 35 degrees north, the California coast takes a little knee. And that knee is called Point Conception. And you'll notice that the warm water part of the California coast goes up to about where Point Conception is. Right where Point Conception is where that cold water coming from the north on the California current is deflected away from the coast, and warm water circulates up from the south. So there's a lot of cold water north of Point Conception, and there's warmer water south of Point Conception. So Point Conception marks a barrier between what we call biogeographic provinces because of the way that water is circulating. There's colder water north of Point Conception and warmer water south of it. And there's relatively few clams and snails that can live on both sides of Point Conception. Most species are going to be adapted to live either in warmer water or in colder water. Not all of them are going to be adapted to live in both. And Point Conception is right at 35 degrees north, which happens to be one of those break points when we look at the biodiversity indices. Right? 35 north is the southern boundary of that clump of shaded blocks right in the middle. Between about 35 and 55 north, those blocks tend to be similar to each other more than they are to blocks outside of that range. And that's why 35 to 55 north corresponds with the area where the North Pacific current is hitting the coast and turning into the California current. And so Point Conception, the boundary is right where the boundary is between the area numbered 15 and 16 on this map. It's a natural boundary between water bodies of different temperatures, and so relatively few species live on both sides. And so Point Conception, California, marks the boundary between two biogeographic provinces. To the north, numbered 15, is what we call the Oregonian province. And province just means an area of ocean with a lot of species in common. To the south of Point Conception, we have the Californian province. South of the Californian province, province number 17, I guess that's the Baja Californian province, and then 18 is the Panamanic province, and so on. There's about 30 provinces that you can divide the shallow oceans into, and all that means is an area that tends to have distinctive water conditions and groups of species that are more similar to each other than what you find in other provinces. They're just areas that have a lot of species in common. Over on the East Coast, you have something similar. This is the East Coast of North America. I can see Manhattan Island. Down at the very south, there's Delaware and Chesapeake Bay. This goes up at the upper right. You've got the state of Maine. And kind of sticking out in the middle, you've had this area of land shaped like a hook. That's Cape Cod, Massachusetts. And that also forms a boundary between relatively warm water to the south and relatively cold water to the north. And that is also a boundary between two different marine provinces, two different areas that have distinctive sets of living things in common. Even in the open ocean, current systems create barriers to dispersal. In 2017, they ran an analysis of 65,000 species of animals and plants that were found living in 5-degree blocks of oceans. And computer analysis found there's 30 different biogeographic provinces in the oceans, 30 different realms. You can see that Oregonian province corresponds to area number 7. I guess that's Oregonian plus Californian. And then 12, is the Panamanic province going all the way down to Peru. 11 is the Gulf of Mexico and the Caribbean, and extending on up towards Cape Cod, Massachusetts. That's going to have relatively warm water, and so on. So even in the completely open ocean, you can find barriers to dispersal, and you can come up with criteria for dividing the ocean, the open ocean and the coastlines into regions that have distinctive sets of animals and plants, biogeographic provinces. Okay, right. Hope that was reasonably clear, and I'm going to go ahead and stop recording.